So then now, I'd like to invite to the stage our keynote speaker, Dr. Kelly Parado, Director of R&D and Bioprocess Engineering at the Human Health Therapeutics Research Center at the National Research Council of Canada. So Dr. Parado has been the leader in advancing in Canada's biomanufacturing and health innovation ecosystem, translating scientific discovery into real-world impact. So today she will be sharing a talk titled From Innovation to Impact, Biomanufacturing Strength and Opportunities in Canada. And then I'm gonna switch to... Thank you.
Great. Okay, thank you so much, everyone. Thank you, Eugene. Thank you to all of the organizers for inviting me here today. I'm very passionate about this topic, so I'll try to keep on time. I do tend to go on. I want to emphasize how timely this subject is around talent development. I spent the last 2 days in meetings focusing on all the growth that's happening in our biomanufacturing ecosystem in terms of new facility builds and rapidly growing companies and new partnerships that are bringing new technologies to Canada. And really the time is now to be accelerating as much as we can the development of talent for meeting this really rapidly growing industry and this important economic lever, I would say, in growing value in Canada. So my name's Kelly Parado. As mentioned, I'm currently the Director of R&D for Bioprocess Engineering at the National Research Council. I am a card-carrying immunologist, PhD trained. I spent my early part of my career in academia developing cancer therapeutics to treat cancer, so oncolytic viruses specifically. Working at the OHRI, and very quickly realized that once you have an exciting discovery and you want to move that into clinical trial, of course there's this chasm of activity that needs to happen, and all of this opportunity awaits on the other side. But it's very challenging to get through that transition. And so we've lived that. We've built on-site manufacturing at a very small scale for our own products. And then I went on through subsequent stages of my career to work in consortia and not-for-profits that were really around linking technologies with biomanufacturing capacity and other investment strategies so that they could grow in these very early stages and start to experience value creation. So in about 2019, I had the privilege of joining NRC. I launched one of the first of its kind challenge programs around cell and gene therapy. The point of the program was to allow government partners like NRC to collaborate broadly with the ecosystem to advance technologies to make cell and gene therapy more accessible and affordable in Canada. And of course, much of this activity focused on manufacturing type technologies and innovations that would drive down cost of goods and democratize cell and gene therapy. So it's with this lens and this vantage point that I'm coming to you. I've spent my career developing and supporting the development of technologies that all hone in on this one point of biomanufacturing and that requirement. And so it's with this view that I'm going to speak to you today. It's obviously the future of biomanufacturing. It's a very large topic. I'm going to try to stay on to several key themes. Really to highlight this critical role of biomanufacturing in therapeutics development and in fact economic development and value creation in promising therapies. I'm going to give a little bit of an overview of the Canadian ecosystem strengths in this area and some highlights on the federal strategy for supporting life sciences through biomanufacturing. That comes from my counterparts at ICED, but they've shared some material with me that I can share with you. I'm also going to talk about biomanufacturing activities at NRC because I think, first of all, it's where I come from, but it's also a really important value chain lever that is actually available to all of Canada's innovations where we have a technology match and complementarity and we can support moving those innovations. Also an interesting, as I'm listening to Eugene speak this morning, it's also going to be probably an interesting place to offer experiential training. And we do this regularly, but we haven't done it as extensively as we would like to. And then again, I'm going to focus on talent development gaps that I can certainly see from my perspective and some of the opportunities, some of which you've heard today and a few others that I've found in my research. So biomanufacturing, of course, is actually extremely vast as a discipline of science and as in application in various fields of the economy. It touches biofuels, bio-based chemicals, industrial enzymes and product intermediates, nutraceuticals. We're, of course, going to talk today mostly about biopharmaceuticals, such as vaccines, monoclonal antibodies, viruses, gene therapies, et cetera. But what they all have in common is the use of biological systems to produce commercially viable goods. And since we're producing biological goods from biological systems, this is inherently complicated when you're then working in manufacturing in a regulated environment. And you have to characterize all the points around your product that you're making and the process that you use to make it. And this complexity actually is a really important contributor to that chasm of development that I sort of talked about between that very early discovery stage and the later stage value creation and a growth of your product and the company that's perhaps advancing it. So this is what we refer to. This is one of probably 100 figures that you could find on the internet around the Valley of Death. This is really at least a first valley of death in any biopharmaceutical product's journey, which is that transit from the laboratory discovery into a phase 1 or phase 2 clinically validated product. And this is that part of the journey where the investment that you would normally have from, say, government grant sources and very early venture capital is winding down as your IP is becoming very interesting. You have some preclinical evidence. But then there's this drop-off where the product is not yet de-risked sufficiently for significant investment or for uptake from the private sector. And to get across that, to actually de-risk that, you have to go through biomanufacturing. You need to go to a phase 1 or phase 2 clinical trial, depending on the nature of your product. And so this is, again, where I think there's an animation here. Yeah. So this transition is extremely expensive and requires very specialized expertise. That's where you'll come in, and facilities. So this is actually some information that some of my business development colleagues have developed at the NRC. We actually, because one of our jobs is to work with the ecosystem to find technologies that need support, de-risking and creating value and moving along that value chain. So we've looked at the Canadian pipeline for biologics, and we've sort of assessed, how many at a given time are sitting at the discovery stage, how many have moved to preclinical, how many then transit to phase 1, phase 2, phase 3 clinical trial. And if you compare that to the global pipeline, I think what you can see is that our drop-off of conversion from the preclinical to the phase 1 space in Canada is about twice as steep as the rest of the world. So we're leaving a lot of technologies on the bench that we're not able to convert into those phase 1 de-risked products. And I think that sounds alarming, but it's something that we know, and I actually think that it's a really important innovation opportunity. And it's not just important because we're leaving technologies languishing or because we're losing technologies from our marketplace. Because we're not advancing these technologies, we're not able to further their value creation. We're also not anchoring and growing companies. So our economic footprint around these technologies is not having a chance to grow beyond that preclinical space where we're losing access to our IP. So again, I think some of the analyses that my colleagues at NRC have run with some of this difficulty with transition at that stage, and certainly not unique to our own findings. But biomanufacturing is actually very critical in this. Access to the expertise, as I said, access to the facilities. So this same group have done some deeper analysis trying to unpack what's happening in the global pipeline in terms of what are different economic and life science regions doing to support their life science ecosystem. Where are they investing and where are they growing? And so it's a very sophisticated analysis that they've done. It was published online in Bioprocess International. I've left the reference here on the slide for you. I'd encourage you to go look at it. It's quite interesting. But what I wanted to pull up was just one quick summary where this group has evaluated various countries, I think 24 different countries, value creation strategies in life sciences by relating total manufacturing capacity relative to their GDP and cross-referencing that to their innovation pipeline capacity against the biomanufacturing capacity that they have. And so they've normalized this as well for the nation's GDP, so you can compare countries like the US and China to small countries like Sweden and Canada, et cetera. And so they've pulled out several trends. You can see sort of in the orange circles on the vertical axis, Countries that have really invested strongly in biomanufacturing and well in excess of their innovation pipeline, they export this capacity. They have business to do manufacturing, contract manufacturing for biologics wherever they come from. And that's actually a line of business. It's a contributor to their nation's GDP. If you look along the horizontal axis, and the further you move to the right on that axis, these are countries like Canada, like the UK, who invest very, very significantly in that early R&D stage. But we have way more innovation than we have historically had capacity in biomanufacturing to convert those technologies. So that's at least one finding from this paper. Again, I'd encourage you to go and have a deeper look, but I think it just puts some numbers down behind some of the trends that we've all been aware of in life sciences for quite some time. And so this inability to convert our early stage biologic innovations into later stage marketed products is not new, but this became extremely evident during the pandemic. When suddenly we had a crisis where we had a number of very fantastic vaccine and therapeutic candidates coming out of Canadian universities and SMEs. The government was looking to fund these, did fund some of them, but there was nowhere to domestically manufacture them at a scale, at a speed, in time to make use of them in our own country during the pandemic. And that was mirrored by a lack of capacity also in large-scale biomanufacturing to secure vaccine supply for Canada. So we ended up, of course, in a situation where we were brokering deals with other countries and major pharmaceutical companies, to procure and access vaccines, but we could not return any kind of in-kind gesture by participating in manufacturing campaigns in a timely fashion. And so this was certainly, I will say, the impetus for the biomanufacturing and life sciences strategy. It recognizes the decades-long history that Canada has in innovation. Many of you are evidence of that in this room. I was hearing all about the statistics around the successes around this ecosystem. But how do we grow those? And so the biomanufacturing life sciences strategy that was launched from ISED is sort of a holistic view of how do you enable innovation. It isn't just about building factories. It is really about how do you make investments in a strategic way across the ecosystem that can contribute to that value chain so we can grow more technologies at scale, and we can get more of those exciting innovations, I'll say, at bat. I was going to say off the bench, but we'll say at bat. And so I'm going to go through just a few of the update slides that I have from my ISED colleagues around the investments that have been made under this strategy. I'm just going to talk about the middle three pillars because they touch more concretely on biomanufacturing. So this is sort of pillar 2. This was their initiative to help to advance the next generation of technologies by strengthening the research ecosystem. So this was really, I don't need to reiterate the creation of the CBRIF hubs and the companion BRIF investments, but the goal here really was to have the ecosystem self-assembling around critical challenges in biomanufacturing, value creation, pandemic response, at speed and at scale, and looking for what the best and the brightest have to offer as solutions and making sure that they have the capital in order to make these successful and put these technologies in the hands of industry. So this was really where the genesis of the 5 hubs have come from. Each hub has its own identity, and there are, of course, important themes that transverse through the hubs, and training, growing the next generation of talent is a very important one. The pillar 3 was about doubling down on existing research capacities and assets of importance. That's how I'm interpreting that pillar. And so really looking across Canada's life science ecosystem for leading universities, research institutes, incubator facilities, pre-existing research networks, and of course, key strengths within the hubs in a targeted way to actually look at, what assets do we have that could grow quickly and help in a pandemic response or add value to the ecosystem in a very rapid fashion. And so again, this was really just another way of recognizing what could be useful and timely and stepping on the gas with capital. And so this was a very important approach because we know that access to capital is very challenging, and speeding things up with cash is extremely essential in an urgent response. And then the pillar 4, as I said, I was talking about this for the last couple of days. Pillar 4 was about extending existing biomanufacturing capabilities and adding new biomanufacturing capabilities. This is bioreactors, this is bricks and mortar buildings, this is investing in facilities that already had some biomanufacturing capacity and could supersize it or could orient their offerings from, say, small molecules and bacterial synthesis into mRNA LNPs. Many of these investments went to companies that manufacture their own products and giving them greater capacity to do so. Some of these investments went to facilities that are like CMOs and CDMOs who will manufacture on a contract basis whichever products come through their doors. And so, again, there's been a lot of tremendous growth. We're hearing a lot of growth, a lot of investment. And so I will talk about the importance of these investments working together. And I wanted to focus on the BMC Inc. and the NRC CTMF. So this is home to me. The NRC received 2 different capital investments during this, the execution of this strategy. One was to build a phase 3 and commercial market-ready facility for the manufacture of nearly approved or to be approved vaccines, or actually licensed products. And the second of those facilities came close behind. This is a phase 1/2 clinical trial material facility for rapid manufacture of new innovative technologies. So just for those of you, many of you might not actually know the National Research Council. We've been around for about 110 years, yet every time I walk in a room, lots of people don't know who we are. So I'll start here. The National Research Council is a federal agency of the government. So we are owned and operated by the government. We are a research performer. We're not traditionally a grant institution. We also don't write much policy, although we do get involved in policy conversations around science and technology and economic development. What we do is we perform research and we maintain facilities and expertise so that we, with our partners, can together advance scientific and technical knowledge. We can orient ourselves to support government policy objectives, so rapidly pivot to respond during a pandemic. And we traditionally and on an ongoing basis conduct research with academics, with SMEs, medium and large companies to support business innovation and provide technology solutions. So that's true for the entire NRC collectively. Our mission is, I'm just going to read it because it's actually a new updated mission statement, is to have an impact in Canada and in the world by advancing knowledge, applying leading edge technologies, whether they're ours or those that come from the ecosystem, and working with innovators to find creative, relevant, and sustainable solutions for Canada's current and future economic, social, and environmental challenges. So that is our sort of driving purpose. NRC has 12 different research centers in different scientific disciplines. That spans what we call planes, trains, and automobiles, so aerospace, automotive and surface transportation studies, quantum and nano, astronomy and astrophysics. So it goes on. We're extremely diverse, and I'll get into my own research center in a moment. But the NRC has gone through a 5-year strategic planning cycle. We're sort of early in the cycle still. But for the 24 to 29 period, we have 3 main pillars of focus, which is to focus all of that effort, focus all of that impact into climate change and sustainability-related technologies, quantum and digital technologies, health and biomanufacturing. So this is really an important and enduring part of our strategy now and into the future. And that health and biomanufacturing does touch outside of the biopharma space. It goes into medical devices as well. It goes into agriculture and crop resilience. But again, at the end of the day, biomanufacturing is absolutely critical to sustaining health and security for Canadians. So it's a really important driver for everything that we do. My research institute is the Human Health Therapeutics Research Center. We're the second largest at NRC. We're about 300 scientists and staff. And we work together to transform human health outcomes for the benefit of society. And we do this by working very collaboratively with public and private sector organizations to accelerate the development of innovative biologic medicines, health solutions, and vaccines in Canada. And what this looks like to us is actually being a co-development partner as needed for these emerging biologic technologies, walking along that value chain, getting across that value of death, moving from the discovery and design phase to apply the science and the expertise and the development capabilities to go to lead candidate evaluation, product and process development, and then eventually scale up for GMP manufacturing for clinical trial. And what I think is really great about this model is that it's the government dollars are sustaining all of this expertise and all of these facilities so that SMEs, academics don't have to do this. You don't have to equip yourself with this magnitude of ongoing expertise and facilities. You can use it collaboratively, come in our doors, and we'll work on a project with you. And while we are doing activity to develop someone's product and get across that valley of death and into that phase 1 clinical trial or beyond, we also work with larger scale partners. We are not just customizing a solution for those products, we're also learning from this journey and saying, oh, okay, we need a tool here. We actually need to do this much faster. We need to do this much better. So we reinvest a lot of our internal budget into creating next generation solutions to either license out technologies, which I'll talk to in a minute, or to actually make the work that we do much better, faster, and less expensive. And so the Human Health Therapeutics Research Center is organized actually into a value chain. So we have a department around drug discovery, a department for preclinical testing. My department is bioprocess R&D, so everything from strain engineering to full scale, at pilot scale, manufacturing ready for tech transfer. And the clinical trial material facility is what I focused on in the biomanufacturing life science investment that we received. This is now a clean room facility for doing phase 1 and phase 2 clinical trials. So the people in my department who are developing process technologies walk down the hall and we hand over the process to our colleagues who are going to manufacture. Across the road, and that's why I put this line here, is the Biologics Manufacturing Center. They're literally across the parking lot from us. So the same people can actually walk across the parking lot and help with the tech transfer over to them. And actually all the facilities are equipped with the same scales of operations of bioreactors, except that the Biologics Manufacturing Center sort of works up to a 2,000 liter volume. But everything between the R&D and the clinical trial material facility is hand in glove seamless. So the clinical trial material facility, I wasn't going to give a lot of its specs because I'm not really talking about that, but it is scoped for any mammalian or insect cell expression of basically any biologic up to containment level 2. So we work mostly with antibodies, recombinant and fusion proteins, virus-like particles, gene therapy vectors, viral vectors, replicating viruses as vaccines. What we definitely don't do are mRNA LNPs. We did that we scoped them out as a strategic call because there's sufficient capacity and there were very substantial investments under the life science strategy in mRNA LNP capabilities. NRC is not here to duplicate any of that. We're really there to fill gaps where they exist. But when people come to work at NRC or to have a product supported, they get to access also our proprietary expression systems and cell lines that we've developed. So oops, sorry, I need to go back. Wrong button. So if you're in biomanufacturing for biologics at all, you'll know how important this is. Many virus-based vaccines are made in adherent viro cells. That's sort of a gold standard and very approved from a regulatory standpoint. We've actually managed to adapt them to suspension. So they are very much more scalable, they are higher producing, and at substantially less cost of goods. So this is now in the hands of, we have licensed this technology to a major pharma partner who is evaluating putting that into some of their major virus production campaigns. But this is a non-exclusive license, so we can still give this out to the research community and we can license it out to any company that's looking to make virus-based vaccines. We also have proprietary CHO cell lines and a CHO pool platform that allow us to rapidly accelerate GMP manufacturing under a pandemic circumstance. I say that with the pool platform because we're pursuing a pathway to manufacturing from a transfected pool rather than an isolated clone. And so that's, again, a regulatory conversation to help us to maintain quality of the product, but shorten the timeline in manufacturing so we could respond with any kind of protein, antigen, monoclonal antibody a therapy or passive immunization for a pandemic response in a speedy time. But for normal therapeutic development under normal conditions, the CHO cell line can be prepared under transient or inducible or stable expression, and we do cell line development, et cetera. We also have a HEX cell platform that is proprietary to NRC that we have engineered for high production of lentivirus vectors to be used in CAR-T and other gene therapy applications. We're working on one now for AAVs for gene therapies. So that was just sort of an example of how we sort of take signals from the work that we're doing and the needs to respond more quickly, more efficiently, and try to engineer solutions, and then we offer those solutions to our partners. The way bioprocess engineering works for us, by the time someone has their very impressive molecule, I'm just going to produce, we call this sort of the gene to batch kind of approach to looking at how we look at a manufacturing project. So someone comes with a sequence for an antibody, for example. We're going to engineer the vectors and expression systems so that they are compliant and ready for manufacturing. We're going to go through some upstream process optimization using DOE and look for the optimal conditions to produce the product in a way that it will be easily purifiable and readily scalable. We start very early on a downstream process development strategy while we are in parallel working on scaling all of that up. Before we do that, we have a lot of technology to help us examine scale up and robustness of the clones that we're picking. You could well imagine if you're waiting 6 months to get your antibody clone, because that's just how long it takes, you want to know that by the time you're done that, it's actually going to be scalable in manufacturing. So we're assessing that very early on. One thing I really want to point out, my analytics colleagues would be very upset if I didn't point it out, but this is really for anyone who's developing a biologic. We often have groups, SMEs and otherwise, coming to our doors with their favorite product, but they haven't done a lot of analytical method development, they haven't characterized their product, they haven't done any pre-formulation studies to have a sense of how this molecule is going to behave in something like a formulation buffer. And very often, when we screen them way back here when they first come to us, and find out that the favorite thing that they've just developed a preclinical package around is not manufacturable. So if it's not manufacturable, it's not a great product. So I just really want to encourage everyone in any audience I talk to, really look at your product quality and indicators of manufacturability. We'd be happy to talk to you about that offline, about how to do that. But this whole process basically then gets transferred into the manufacturing facility, the pilot scale, the at-scale process, all of the companion analytical methods, et cetera. We can also work in a way, you don't have to use our clinical trial material facility. We're the government, we don't force anybody to use our tools. If we've made a process, we will transfer it anywhere you want to go. And in fact, we've been doing that for 20 plus years prior to having the CTMF. So that's very natural for us. We also take in more mature processes where sometimes a manufacturer wants to change a component and they want to test it and de-risk it in our environment. So that works as well. And what I really want to emphasize is that we work across all kinds of therapeutic platforms and emerging technologies, things that I've never heard of walking in the door. So every day in this environment is exceedingly interesting. And I think that's going to be important for thinking about how we think about the next generation of talent who's going to be looking at creating and biomanufacturing innovation in this space. So like everybody else, NRC received instructions on how we were to invest in innovation in this strategy. So they really want us to use this capacity in a way that is extremely flexible so that we remain competent, I keep pushing the wrong button, competent and capable regardless of what therapeutic platform, or in this case, vaccine platform comes into our doors. We have to be able to quickly pivot to meet pandemic requirements, and we're constantly innovating in ways to actually shorten the manufacturing cycle and that bioprocess cycle. But at the same time, these investments are not just for pandemic preparedness. We're building all this manufacturing capacity. Of course, we want to be able to use it to support economic development and innovation. And so really, they're wanting us to look ahead as well at solutions for precision medicine, cell and gene therapy, advanced viral vectors that are coming, multifunctional monoclonal antibodies, et cetera, and also to start thinking about leveraging strengths in the ecosystem such as AI to drive health innovation. So we are definitely thinking about this as we're creating our internal research programs and looking for areas to collaborate. And also, I wanted to share with you just a few insights. Probably these aren't going to be surprising, but I thought I'd share it anyway. Because we do meet with clients, industry clients all the time, probably at least 100 or 150 a year, we're having conversations around their various product challenges. So they come to us looking to solve product development challenges, not usually just to provide services, but also to how do I make my molecule manufacturable? How do I make sure it's not aggregating in my formulation buffer, et cetera? How do I choose something that's thermostable, that sort of thing. So we work in this way, again, as I mentioned, varied product types, usually complex. They're becoming increasingly complex. But everybody wants everything fast, low cost, and high quality. That's not a surprise. So we constantly are investing in optimizing the platforms that we already have. We're always working to find ways to streamline and automate our processes. We're starting to use AI tools to help us do this. We work in transiting to more and more scalable systems. So a lot of gold standard manufacturing methods, they are scalable, but they're scalable by replicating bioreactors rather than having a technology that allows you to achieve similar output at a smaller scale. We maintain state-of-the-art facilities. As I mentioned, we maintain this extremely vast and varied expertise so that we can call in the necessary experts when a new challenge arises. And we're always working to facilitate also that tech transfer bridge between the R&D environment and any GMP environment, not just our own. And so just a couple of notes on what we're thinking about in our own internal research programming and where we're starting to look at collaborating. So again, optimizing platforms we already had. I had sort of mentioned working on increasing performance of our current platforms, so getting to greater speed and scale. And we do this through a combination of cell innovation approaches or process innovation approaches. I have a couple of examples. How am I doing on time, Eugene? Right on time? Okay. Okay, so I'll go a little bit quick through these projects because I want to get to the training part. So we're working on methods of accelerating protein production in GMP. I did mention that moving into GMP by using a stable pool approach rather than taking the 4 to 6 months it takes to make a stable clone, but also interfacing with regulators to understand how is that going to be accepted by regulators. We even have the question, will this be accepted by regulators in a non-pandemic circumstance if we can prove it out in a pandemic-ready type response. And cost-effective viral vaccine production at scale, that's our Vero cell line. We have a version 1.0 Vero, which is the one I told you had been licensed and is very scalable. We're now doing a lot of precision engineering and editing in that cell line because we understand certain features of the cell, doubling time and that sort of thing that we'd like to enhance so that we have a superior performing cell line. We're also working on a lentivirus. If any of you are familiar with cell and gene therapy and lentiviruses, traditionally they're made in adherent cells. We've transited this, of course, into a suspension cell, but we've also stably integrated many of the viral packaging genes into the cell line under an inducible switch. So we have what we call a packaging cell line where you only have to introduce the gene of interest to then get your lentivirus. But we're now working on, we have created a first version of a stable producer where everything about the lentivirus is under an inducible switch and you just turn it on and you can scale it to 200-liter production and pop out your lentivirus. But again, we've learned a lot in that first generation, so we're bringing back genome engineering tools to optimize the cell line. We're working quite significantly in process analytical technologies, and so how do we measure what's happening with the cells, the product, and the process in the bioreactor in real time so that we can use this to empower AI models and machine learning to inform the design space for biomanufacturing and get to answers around process control much more quickly. And then moving towards the future, we're sort of looking at getting into continuous bioprocessing as a means to further increase the efficiency and the speed of manufacture, increased controls, AI controls and analytical technologies in manufacturing. Our sister research center, Medical Devices, is working on more microfluidic scale device manufacturing. So my research center thinks of doing things big. They think of doing things very small and increasing the efficiency by decreasing the interface in your biologics in the reaction. So really interesting technologies there. And I know some of the investigators in this hub are actually working with my colleagues. That for us is going to require a lot of investment and infrastructure capital, so that's sort of probably going to be in our next 5-year cycle, but we're always working in a way to increase speed, efficiency, and quality of what we're doing. So in the interest of time, I'm going to speed through. I was going to go through like the projects I just summarized. I'm just going to speed through the couple of slides because I don't think I need to go into any of the specifics of those projects. I think the impact was probably the most important point. Oops, apologies for all of the animations. Oops. We're almost there. Okay. So The NRC, like I said, the reason we do this, we're not a commercial manufacturing organization. We're not doing this for money at all. For us, success is when we create value for the company. So that means advancing technology that doesn't belong to us. It means validating someone else's technology and increasing in parallel the valuation of the company, helping the company to attract increased investment, helping that company to grow and add jobs to themselves, and help them and all of us contribute to the ecosystem. So those are our outputs, those are our metrics. It's not sustainability of our budget, it's creating value for Canada. And at the end of the day, we exist and we do all of this work and activity so that we can help to introduce more technologies into that phase 1-2 clinical evaluation. When I was at a talk yesterday, I don't have the footnote here to cite the reference, but the keynote speaker there indicated that from 2020 to 2024, half of the therapies that were approved by the FDA started in universities. That is significant. So, and those are approved therapies, so you can imagine how many also failed along the way. So we really need to do a great job of getting therapies at bat. Oops. So just two more notes. The NRC doesn't do this alone, of course. We collaborate quite broadly. The Government of Canada had formed an MOU with the Government of the UK, put out several advanced manufacturing calls. NRC is a collaborating partner. The goal of this really was to demonstrate Canada and the UK's shared commitment to innovation and to use our innovation ecosystems to try to drive innovative solutions around complex biological therapies. We've been involved in, I think, about 7 different proposals that have been funded. I can't announce them because they're not all signed and they're not public yet. But everything from creating new AI tools around manufacturing to and automated biomanufacturing devices, point-of-care biodevices, that sort of thing. And of course, we also work with the hubs to varying degrees. We have, again, with this value chain that we offer, the ability to help move anybody's therapies and guide your developments, thinking for success, what is the end in mind for the product and innovation, and how do you want to get that into the hands of a partner? So we're available to work. We also are available to host trainees, as I was sort of alluding to earlier. We're not a degree conferring institution, so we don't tend to create training programs. We're always happy to participate and provide some experiential learning type environment, and I'll give you an example of that. So my last few slides, really going to touch on talent. So this is an article that appeared in the Globe and Mail, co-authored by a former NRC colleague, actually, Dr. Amin Kamen. Really showcasing, like really taking a step back and looking at all this growth in biomanufacturing facilities, all those companies on that map that I showed you that got all these great investments. Whether they're manufacturing their own therapeutics or everybody else's therapeutics, they need people. And the ability to have individuals trained who are flexible, who are knowledgeable about several areas of technology, because this field is expanding and becoming increasingly complex. The biologics coming forward are increasingly unique, so too are the tools that we have at our disposal to actually make them effectively and quickly. So I can give you an example of what NRC looks like in terms of what kinds of people we have, what kinds of jobs they're doing. So in the process engineering department, way down here, so we're one of 5 departments in this research center. So this is where we do all the platform engineering, that includes microbial strain expression, but also mammalian. Process development and scale up, all that analytical, all the formulation development that I was talking about. So these individuals, they come to us usually with traditional university training, so in biotechnology and engineering, deep in cell and molecular biology, genetics, bioengineering, chemistry, biochemistry. The teams themselves tend to be extremely cross-functional and multidisciplinary. So I don't have, my cell culture team is not just people who do cell biology. They are chemists, they are engineers, they're physicists working in this team. So staff themselves, each individual also tends to tends to master multiple disciplines themselves so that they can work with other teams seamlessly and also interface with clients and collaborators. I keep saying this, AI and automation, well, they're coming to the NRC, they're already here in industry. And so I think that individuals who have that ability to interface between digital technologies, AI and machine learning and biology, I think are going to be extremely vital in this industry going forward. And I think that in addition to industry placements, because many of our staff have come from industry, seeking opportunities for interdisciplinary training and manufacturing specific theoretical or hands-on training is actually going to be very essential. And I'm giving you just one example where we do participate. McGill University has a Master's of Engineering in Biomedical Engineering program. It's sort of 8 months or a year of course curriculum. Much of this curriculum is taught by scientists at the NRC. And then 8 months experiential internship. And the McGill University program has a network of industry partners that can actually host trainees. NRC is one of those partners, so we have brought trainees from this program to be interns with us for 8 months, and I think many of them have actually been hired in my department. It's been a really valuable experience. They actually work anywhere on that value chain, so some are doing formulation and some are doing bioreactor scale-ups to 500 liters. It's actually really a great environment for them to do that. We'd be very happy to participate with other hub networks and their programs in this way. And again, I just sort of noted, Eugen had shared some of the information about your training programs, and some of ears perked up around your knowledge exchange program, and just supports and opportunities to really have trainees working on multidisciplinary projects or have experiences in environments, in labs, developing technologies from various viewpoints, I think is going to be very important. The HQP that are in our GMP facility, it's quite a different setup. The teams are, so I will say the people who work there, again, have industry experience, many of them, but traditional university or college training in sort of biotechnology and engineering related disciplines. These teams are highly focused, so they're not multidisciplinary themselves. They may have that in their background, but their job, very highly focused, extremely precise, very detail oriented. But it's everything from executing a process to managing the mechanics in the facility. It's extremely complex, all of it's regulated. And so looking for opportunities for training in clean room skills and engineering under GMP conditions is also really important to get a sense of what it's like to actually do scientific work inside a facility. It's actually really challenging. It can be very rewarding as well. So again, I was sort of perking up at the opportunity for industry internship opportunities to get a vantage point from some more mature placements in more mature industries who are doing a lot of these activities on a regular basis, where you can actually look at product development from not just the lab operations perspective, but also from the medical affairs. How are you going to deploy that therapy into medical centers? QC, QA is extremely important throughout the process. Engineering, I can't say enough about the importance of engineering and biomanufacturing, so that's another area to look for opportunities. And manufacturing clean room training programs that I've certainly been aware of. So I know that you're already partnered with Castle and with CADI, and I think those are absolutely fantastic. Another one I just wanted to bring up is one called CanPrime. It's been born out of the BioCanarex NCE network. They also offer MyTax-sponsored experiential learnings and a 2-month micro-certification course that's offered through Algonquin College for clean room work for HQP. So I think that they've trained about 40 or 50 HQP by now in a network of clean room sites at universities across Canada. So that's another one. They just happened to be at some of the meetings I was at the other day, and they're really looking forward to welcoming more and more trainees all the time. And again, the opportunity to look. I love that you've put together, BioHubNet, this program around looking across the various activities in that value development. So, knowledge about the industry. Biomanufacturing, of course, we've talked at length, but regulatory affairs, the design of clinical trials, and actually having manufacturing developers talking with or understanding the needs from the clinical implementation perspective and integrating that into the process design is very, very important. So too is understanding the business and commercialization path and how they are intertwined with the stages of development of your therapy and what sort of evidence you need to bring forward to get that next round of investment, how you need to present yourself, I think is extremely important. So I just want to commend the BioHubNet for how comprehensive a program that they've put together. This is my last slide. So looking to the future, I think we've talked at length. Canada has made very, very comprehensive investments in the life science and biomanufacturing ecosystem. we think we're going to be ready when a pandemic comes because we've got all this capacity. But I do think there's actually a lot of work to do because, having the facilities, we're now going to have to equip them. Obviously, that's why you're here today to learn about that. We also need to connect those early stage technologies with the facilities. There's still more work to do. A word that I heard over the last 2 days is risk capital. Where is money going to come from to actually do all of this manufacturing investment at that early stage to get to the clinical trial step? So I think that there's work from some of the policymakers and the thinkers in government and elsewhere to understand how can the ecosystem access that risk capital and under what conditions. Because really we need to make sure that we're not just discovering, we're not just doing a phase 1 trial, we are commercializing, we're building businesses, we're growing businesses, and people along with that. People are vital to all of this. I also think that as these more complex tools in biomanufacturing are coming to light, we also need to understand how to come together to ensure that they are sufficiently diffused and de-risked from a regulatory standpoint. So I was in a talk yesterday, thinking again about the idea of using AI to control your biomanufacturing process based on information that is coming from the real-time biomanufacturing process. How are regulators going to see that when they get a regulatory submission around the manufacturing at the end? We don't know yet. So I think that it actually is going to take a bit of a critical mass of individuals with expertise in that type of space to actually put together submissions and to start talking with the regulators to figure out how can these innovations actually be implemented. And lastly, these facilities, again, have such great potential. All this capacity has potential to orient to pandemic preparedness, economic growth. There are new geopolitical challenges that we're facing too, so these instruments and all of this talent can be pointed into areas like gaps in the supply chain, other health priority areas like access to high-cost drugs for rare diseases and biosimilars. So I hope I'm leaving you with some excitement. I'm always excited about this topic. I could talk a lot more. I could meet you for coffee and the networking and turn over the control. For now, I'd be happy to answer any questions.
I think we still have time for one question. If there's any question in the audience, please put your hands up and then.
One over there.
Oh, there you go. We have a mic ready.
Hi, I'm Nazish. I'm from Biomanufacturing Solutions. We are a pre-seed and also growth industry sponsor, right? We help startup companies. What I see from the startups who are going to be the next decade of Canada and if not globally, I don't see that on NRC's radar, which is personalized medicine. Mm-hmm. And you mentioned that is out of scope for all the growth activities that you're doing, which are phenomenal, and thank you for that. But I do want to comment on personalized medicine.
Yeah, I can absolutely comment on personalized medicine. So what I would say is from the manufacturing perspective, and I wanted to contain it to that, from the manufacturing perspective, we're not going into something like autologous cell therapies. Because again, NRC has a very specific mandate not to compete with industry. And where there's sufficient capacity, we should not be there. We should not spend Canadian government dollars doing things that industry is already doing. So we're really trying to hone down and focus on critical gaps. There's CCRM, there's Omnia Bio, there's all kinds of facilities and companies that are doing this sort of work. But we do work with them in terms of the innovation and analytical strategies. We have projects looking at metabolic profiles of CAR T cells as they're being manufactured, trying to understand how that relates to performance. So we do innovative support work, but we don't do biomanufacturing in those spaces. Same thing with mRNA LNPs, personalized or otherwise. There is more than sufficient capacity. We really felt that we'd be catching up more than we'd be helping in those spaces.
Thank you so much.
Okay, great.
Move back. So thank you, Kelly, for that insightful keynote. It's great to hear about the strength and the opportunities in the Canadian biomanufacturing space. And again, there are so many partners that you have shared today and how we can all work together and collaborate to cultivate the talent and also to drive Canadian innovations forward.